Hello everyone. Welcome to Genomics and Proteomics in Action, the podcast where we explore molecular biology, help us better understand and protect our environment. I'm Amir.
And I'm Daim, and today we'll be discussing how functional genomics and proteomics contribute to environmental monitoring, pollution assessment, and also bioremediation. And I'm Hazik, and today we will talk about microbial communities, biodiversity, stress indicators, and how they all link back to genes and proteins.
And I'm Shafiq. We also cover how these tools are applied in real-life conservation and remediation effects.
Okay, let's begin by dividing into the world of functional genomics. Now, this field goes beyond simply knowing the DNA sequence of an organism. Instead, it aims to understand how genes actually function, how they express, and how they interact with each other. And most importantly, how this interaction change in responses to environmental stresses like pollution or climate shift. Unlike classical genetics, which might focus on one gene at a time, functional genomics actually take a system-wide approach. It looks at the bigger picture, thousands of genes working together within the context of the whole organism or even an entire microbial community. And one of the most powerful tools in this field is metagenomics. Metagenomics, instead of isolating and culturing individual microbes in a lab, which, let's be honest, only works for small fraction of microorganisms. Metagenomics let us extract and sequence all the genetic material directly from an environmental sample like soil, river, sediment, or wastewater.
So, wait, just to be clear, you are saying we can study entire microbial communities without ever having to grow them or even see them under a microscope.
Yeah, exactly. That's beauty of it. Traditional microbiology methods miss out on more than 99% of microbes because they simply can't be cultured. But metagenomics bypass that completely, and we are talking about DNA sequencing from the source, the environment itself. So it's like give us an incredibly detailed to look at how microbes function in ecosystem. For example, in nutrient cycling, like how nitrogen or carbon is processed, or in breaking down harmful pollutant like oil, pesticide, or heavy metals, we can actually see which genes are active and what enzymes are being produced in real-time condition.
Oh, that's kind of impressive though. So, let's say if we're dealing with something like contaminated soil from an industrial site, we could use this data to pinpoint which microbes that are doing the cleanup work.
Yeah, absolutely. In fact, that's one of the major application of functional genomics in environmental conservation is bioremediation. So bioremediation, by identifying key genes responsible for detoxifying pollutant, scientists can tailor microbial consortia. So basically, it's a microbial teams that are best suited to break down specific contaminant. And it's not just academic. Government and environmental agency are already using this data to guide clean up effort in oil spill, agricultural runoff, and heavy metal contamination.
So it almost sounds like we're building a microbial SWAT team because by sending it in the right organisms with the right genes to fix a problem.
Okay, that's actually a great way to put it. So, precision ecology. We are no longer guessing what might work, and we are applying genomic evidence to restore balance in ecosystem.
Oh, and I'm guessing this approach also helps us predict the environmental issues, right? Like catching problems before they spiral out of control.
Yeah, exactly. That's one of the biggest advantage. Functional genomics offer what we call molecular levels, early warning system, before any visible damage happen. So before fish die off, crop fail, or algal bloom explode, we can detect a shift in gene expression or microbial composition that signals something's wrong. So, which means that these early detections means intervention that can happen sooner and with greater precision. And in a world facing climate change, rising pollution, and habitat destruction, that speed could make all the difference.
Okay, thanks, Amir. Building on your points, I think it's very important to emphasize just how central microbes are to ecosystem health. These tiny organisms may be invisible to our naked eye, but they are also responsible for huge environmental process, from breaking down organic waste and detoxifying pollutants to regulating the nitrogen, phosphorus, and carbon cycles. They are basically the nature's engineers. Wow. But here's the catch. Many of these microbes, maybe more than 90%, can be cultured in the lab using traditional methods. We just don't know how to grow them under artificial conditions. And that was a very big limitation for researchers for decades. But now, thanks to the functional genomic tools like metagenomics, we don't need to grow them. We can sequence their DNA directly from the environment, soil, water, and even air. It's like getting a snapshot of all the genetic activity happening in an ecosystem.
But how exactly do we use that kind of information for biodiversity conservation?
That's a very great question, Shafiq. This is where techniques like DNA barcoding and environmental DNA or eDNA really come into play. Let me break it down one by one. DNA barcoding involves sequencing a small standardized region of an organism genome. It's kind of like scanning a supermarket barcode to identify the species. And then with eDNA, we can take that concept even further than that. We can collect samples from water, soil, or even the air around us and find the traces of DNA that animals and plants leave behind. This could come from skin cells, mucus, hair, or feces. We don't even need to physically capture the organism to get it.
So you're saying even if we never see or catch the animal, we can still tell it is been there?
Exactly. And this is a game changer for conservation biology. eDNA is now being used to detect rare or endangered species like the Chinese giant salamander, to monitor fish population in rivers and oceans, and even to track invasive species like the zebra mussel and before they take over an ecosystem. And what's really powerful is that this method is not invasive and we are not disturbing any fragile ecosystem or stressing out the wildlife just to get the data. And that means eDNA is ideal for long-term monitoring across large or remote areas. But there's a catch. The power of genomics doesn't stop at just identifying the species. It also helps us understand population genetics, things like gene flow, genetic diversity, and we can use genomic data to figure out if two tiger populations in a different forest patches are still interbreeding. That kind of insight tells us whether a habitat corridor is working or if we need to step in with conservation action. And other than that, it also gives us insight into evolutionary relationship. By comparing genomes, we can determine how closely species are related, whether they have undergone recent adaptation, how much genetic variation exists within a population. And all of this that I've said is a very important factors in designing the protected areas that actually preserve long-term genetic health. And if you guys want an example, let's take coral reef. Genomic studies are being used to identify coral strains that are more heat resistant and that's critical in an era of rising ocean temperature. Conservationists can then prioritize those resilient strains in reef restoration effort. So overall, genomic is transforming the way that we understand biodiversity, different from what we have learned. It helps us move from simply counting the species to truly understanding how life interact, evolve, and survive in complex ecosystem. And the more that we understand, the better equipped we are to protect this ecosystem. not just now, but for the generations to come.
Alright, we have talked a lot about DNA, genomics and metagenomics, right? But now let us shift gears and talk about what happens after the genetic instructions are read, enter the world of proteomics. While genomics shows us what an organism can do, its blueprint or potential, proteomics tell us what it's actually doing at any given moment. It is the study of proteins, which are the workhorses of the cell. Everything from metabolism, stress response, defense mechanisms to growth, it's proteins making it happen. So, in a way, proteomics gives us real-time information. It is like having a stethoscope pressed to the heartbeat of an ecosystem. We are not just looking at what genes are present. We are looking at how organisms are reacting to it right now, to their environment.
So it's like checking how an organism is feeling rather than just knowing what it's capable of.
Exactly. That's a great way to put it, basically. For instance, imagine a fish swimming in a polluted river. Even before we see any physical symptoms like lesions or changes in behavior, we can often detect elevated levels of stress response proteins such as heat shock proteins, HBSN for short, or metallothioneins. I don't know how to pronounce it. So these proteins are like emergency responders. They get expressed when the organism is under oxidative stress or when it is exposed to toxic metals like mercury or cadmium. So we can use these proteins as biomarkers, early warning signs that something in the environment isn't right.
That's really useful for early intervention, right? Especially if we want to stop the environment damage before it gets worse.
Exactly. The earlier we detect stress in a population, the faster we can take action. Whether that means changing water management policies, limiting pollutant discharge, or improving habitat conditions. And the beauty of proteomic analysis is that it can often be done non-lethally. We don't need to sacrifice the animal. A small tissue or blood samples supposedly enough to assess hundreds or even thousands of proteins. That's incredibly important when we are working with endangered or threatened species where every individual counts. Now, in terms of technique, one of the most powerful tools we use in proteomics, do you guys know?
I don't know.
Okay. So basically, it is mass spectrometry.
Okay.
It is a bit like a protein detective. It identifies and quantifies thousands of proteins in a single run, showing us not just which proteins are present, but also how their levels are changing under different conditions. So these kinds of molecular profiling is now being applied in ecosystem health monitoring. For example, scientists studying coral reef can detect stress proteins associated with heat damage before coral bleaching occurs. Or in forests, proteomics is used to understand how trees respond to drought by examining proteins involved in water transport and leaf development.
So I guess these protein patterns can help in guiding the conservation strategies too, right?
Absolutely, Diane. That's one of the most exciting parts. Proteomic data can reveal which populations are under the most stress, which environmental conditions are triggering that stress, and whether current conservation efforts are actually helping or not. So let's say we are trying to rehabilitate a wetland. Genomic analysis might show us which species are present, but proteomics can show us whether those species are thriving or just barely surviving. Are they producing growth proteins? Are their immune response stable? That's the level of detail we get. And because proteins respond quickly to environmental changes, proteomics helps us to track trends over time, how ecosystems respond to pollution reduction, climate stress, or restoration efforts. It is not just a snapshot, it's like watching the heartbeat of the environment evolve in real time. So in short, proteomics adds a vital dimension to environmental science. It helps scientists or researchers, conservationists, and policymakers make decisions based on real biological responses, not just assumption or visual signs. And when you combine proteomics and genomics, you get a complete picture from genetic potential to functional reality.
Okay, so far, we have explored how genomics and proteomics reveal the inner workings of organisms and ecosystems. Now, let's look at how this knowledge is being applied, especially in environmental remediation, which is all about restoring polluted or damaged environments, and in conservation efforts aimed at protecting the biodiversity. When we combine the functional genomics with proteomics, we get a complete dynamic view. Genomics tell us which microbial genes are present, meaning who has the potential to degrade pollutants like oil, plastic, pesticide, or heavy metals. But proteomics tell us whether those genes are actually being expressed, whether the microbes are actively producing the enzymes and needed for degradation.
Both are needed. One shows the potential and the other shows performance.
Yes, that's exactly how it is. Because think of it like a sports team. Genomics give us the player roster, all the talents and abilities each player brings, but proteomics tell us who's actually on the field, playing the games and scoring goals. We need both to design and monitor effective interventions. Take oil contaminated soils as an example. In the aftermath of an oil spill, we can sequence the microbial community and identify genes coding for enzymes like alkane monooxygenases, which break down the hydrocarbons. But then, with proteomics, we can confirm if those enzymes are actually being produced in situ under real environmental conditions. If not, we may need to adjust the factors like temperature, pH, and nutrient availability to activate those pathways. And this doesn't stop at the detection of it. These tools are also crucial for monitoring the progress. If a bioremediation strategy is working, we actually expect to see a reduction in stress-related proteins, such as heat shock proteins or oxidative stress markers in the local organism. This indicates that environmental conditions are improving and that the cleanup strategy is very effective on a biological level, not just visually.
If it is like that, what about genetically engineered microbes? The ones specifically designed to clean up the toxic waste? How do we monitor those safely?
Oh, that's a very interesting question. And that's an important point of it, right? When we deploy the GMOs in the environment, whether it's bacteria engineered to degrade the plastic or plants the engineered to absorb heavy metals, we have to make sure they are doing what they are supposed to do without causing harm. The proteomics plays very important role here because it can help us to confirm that the engineered genes are not only present but functionally expressed. And we also need to check for the unintended side effects like the expression of harmful proteins or metabolic imbalance. This helps us to ensure the safety and accountability, public trust in the biotechnological solutions. But beyond the pollution, these tools are just as useful as the conservation biology. Let's take examples such as coral reef, one of the most vulnerable ecosystems to climate change, right? When the coral reef experience thermal stress, they begin to express specific heat shock proteins and other stress markers. By analyzing the proteomic data, scientists can identify which coral species or strains are more resilient to rising temperatures. That's the powerful information because it allows the conservationists to focus on restoring the coral reef with heat-tolerant corals. Similarly, in agricultural and reforestation, the proteomics help to identify the drought or salinity-resistant plants, guiding restoration in degraded or climate-stressed regions.
That really shows how molecular data can guide on the ground conservation with precision instead of guesswork.
Yes, that's absolutely right. This integrated molecular approach using both the genomic and also proteomics, which allows us to make smarter and more informed decisions. It reduces the trial and error and ensures that our conservation strategies are scientifically grounded, ethically responsible, and lastly, ecologically sustainable. We are not just reacting to the problems, we also need to predict, prevent, and also restoring in a way that's faster, more targeted, and also more aligned with the complexity of real ecosystems.
See,
One take kan? One take, tentu nak buat one take.
Kau yang gelak apa macam aku? Aku tak gelak pun.
Satu, dua, tiga.
Okay, that's all for today. Functional genomic and proteomic are powerful tools helping us protect ecosystem by detecting problems early and guiding smart solution. And thanks for listening.
Don't forget to subscribe, like, and comment.